Welcome to today's lesson on cellular transport. Last time we discussed that cells need to maintain homeostasis or a stable internal environment, and they do this in part by controlling what moves into and out of the cell. Today we're going to discuss how that cellular transport actually happens. We know that small nonpolar substances can go directly through the lipid bilayer, but other substances like polar molecules and larger molecules need other mechanisms to get into and out of the cell. There are two main types of transport: passive and active transport. Passive transport requires no ATP, no energy, because substances are moving along what is called their concentration gradient. And there are three types of passive transport: diffusion, facilitated diffusion, and osmosis. Active transport, on the other hand, requires energy. It involves moving substances in directions that are against their concentration gradient. We'll discuss what that means a little bit later. So what is concentration? Concentration is the amount of substances in a given volume, and a concentration gradient involves having an area with a higher concentration than another. We call that a concentration gradient. During passive transport, substances move from an area of high concentration to low concentration or down their concentration gradient. So right here you can see two beakers, and they each have 10 solute particles inside of them. There's 10 in each beaker. However, even though they have the same number of solute particles, they have different concentrations because the volume here is smaller, which means that the particles are more crowded. We say that this beaker has a higher concentration. This beaker, on the other hand, the 500 milliliter beaker, has a lot more fluid and the particles are more spread out. So we would say that this has a relatively lower concentration than the other beaker. The first type of passive transport is simple diffusion, and during simple diffusion, small nonpolar molecules pass through the lipid bilayer along their concentration gradient. They move down their concentration gradient, so they go from an area where there's high concentration to an area where there's low concentration. Substances have a natural tendency to spread out as molecules bump into each other, therefore no external energy is required. Here's an example. If you take a cube of sugar and you put it into a container of water, it could be a teacup, but in this case it's a beaker, it's not going to just stay there in one area. What's going to happen is the sugar molecules are going to start bumping into each other and spreading out, and they're going to move from an area of high concentration, like right here, to areas of low concentration. They're going to spread out from each other until they're going to reach equilibrium, and the sugar molecules are equally distributed in the entire solution. So that's simple diffusion, and this is how substances that are nonpolar and really small can get into and out of the cell. They just simply move along their concentration gradient. The second type of passive transport is facilitated diffusion, and facilitate means to help. So it's a diffusion that gets some type of help. And the help that's needed for facilitated diffusion is one of these transport proteins that we discussed last time. So ions can't just go directly through the cell membrane. They get repelled by the cell membrane, so they need to have kind of like a tunnel or something to pass through to get into and out of the cell. And that tunnel is the transport protein. So facilitated diffusion involves the movement of particles from high concentration to areas of low concentration through a transport protein. And that's what's happening here, because outside the cell we have a huge concentration of these little square molecules. Inside the cell there's a low concentration. These are going to move down their concentration gradient into the cell. And no energy is required because, once again, we're going along our concentration gradient. No energy needs to be added. No ATP needs to be added. The third type of passive transport is osmosis. Osmosis is really the diffusion of water through a selectively permeable membrane like the cell membrane. And selectively permeable means some stuff can get through and some stuff cannot. This allows the cell to maintain water balance, which is an important piece of homeostasis. So water moves through water channels from high concentration to low concentration. And when during osmosis, it's a little bit tricky because we're looking at water. We're not looking at what the particles are doing. We're looking at what the water molecules are doing. So here is an example. In a beaker, we put a semi-permeable membrane in here. So there's like a wall here that water can pass through, but the solute particles cannot get through because they're too big. So in this example on the left, there is a lot of water and a low solute concentration on the left side, and on the right-hand side there's a high solute concentration, but that means that the water molecules are less concentrated. So what happens here is water is going to go along its concentration gradient, and the water molecules are going to move from where they're highly concentrated to where there's a relatively low concentration. Water is going to go down its concentration gradient, and what ends up happening is the water molecules cross over to the other side, and the water level rises during osmosis. So let's discuss osmosis in the body, particularly as it involves cells like red blood cells. So there are different types of solutions that can occur, and the first type of solution is a hypotonic solution. In a hypotonic solution, a solution has a lower concentration of solutes than the cytoplasm of the cell. So here's an example. This is the cell, and it has 20% salt concentration. And the water that's around the cell has a 10% salt concentration. So that's lower than what's inside the cell. In this case, what's going to happen is the water is going to move down its concentration gradient. It's going to go where there's a lot of water to where there's less water. So the water is going to rush into the cell. And what happens is the red blood cells here are going to get swollen, and they might even burst open and pop. So this is a pretty dangerous situation. When you get an IV drip, it's very important that that solution is not hypotonic, because this is what would happen if you put your red blood cells in, let's say, pure water. The second type of solution is hypertonic solution. In a hypertonic solution, a solution has a higher concentration than the cytoplasm of the cell. So here we have 20% outside, and inside the cell there's a 10% salt concentration. In this case, water again is going to move down its concentration gradient through osmosis, and it's going to move outside of the cell because there's more water molecules, higher concentration of water molecules inside the cell than outside. Remember, the blue dots don't represent water. That represents the salt, right? So the water is going to do the opposite of what you would expect those little particles to do. So in this case, water is going to leave the cell. And what happens here, you can see it here on a plant cell, water moves outside of the cell, all right, and the cell shrinks. In a red blood cell, you can see a cell here that has completely shrunken and another cell here. That's not a good situation, right? The cells are going to shrink, and that's not healthy for the cell. So that is what happens to cells in a hypertonic solution. So if hypotonic solutions are not good and hypertonic solutions are not good, what kind of solutions are ideal for cells, and what kind of solution would you get if you had to get an IV at the hospital? And the answer to that is an isotonic solution. In an isotonic solution, the concentration of solutes in the solution is equal to the concentration of solutes in the cytoplasm. So we have a 10% salt concentration outside, a 10% salt concentration on the inside. So the rate of water leaving the cell and the rate of water entering the cell is going to be equal, and there's going to be no net change in the size of the cell. So your red blood cell is going to stay perfectly healthy. The right side is not going to swell, it's not going to burst, it's not going to shrink. So this is the ideal type of solution. So these were the types of passive transport. Let's look at active transport. So one more time, active transport involves processes that require ATP, that require energy, because substances are going to move against their concentration gradients. And there's two types of active transport we'll look at today: pumps and vesicles. Pumps in active transport, carrier proteins require energy to pump substances against their concentration gradient. So we're going to move things in the direction that they don't want to go. We're going to go from areas of low concentration, and we're going to move to areas that are already crowded, that already have a high concentration. And of course, this is going to require energy. Think of this as there being an elevator that's already concentrated and full of people, and you're one of the people on the outside, and you want to move into the elevator. You got to kind of move people over, use some energy to get onto the elevator. That's exactly how active transport works. And there's a very famous or a very important type of pump called the sodium-potassium pump. And this is a carrier protein that uses ATP to actively transport sodium ions out of the cell and potassium ions into the cell. And this carrier protein, the sodium-potassium pump, is really important because it prevents the buildup of sodium ions inside the cells, which could lead to osmosis, causing the cell to swell and burst, as we saw in the previous example. So that's the sodium-potassium pump. And you can see this happening here. during active transport, we're taking particles that are in a low concentration out here, and we're pushing them into areas where they're already in high concentration, and sometimes cells need to do this. Okay? So this is a comparison of passive and active transport. This is simple diffusion, which can go right through the cell membrane. Particles are moving down their concentration gradient from high to low concentration. No energy is required. Here we have facilitated diffusion, and again, we're going down our concentration gradient from high concentration to low concentration. The only difference is we're using a transport protein to do it, right? Because we have something that's polar, for example, and it can't pass through the cell membrane directly. During active transport, now we're moving against the concentration gradient. So if you look at these little squares, they're more concentrated inside the cell than outside the cell, yet we're moving these squares to where they are more concentrated. In this case, this transport protein is going to require ATP to move these solute particles against their concentration gradients. Okay. The last type of active transport involves the use of vesicles. Large substances can't get through transport proteins, and they certainly can't get through the cell membrane, so they need another mechanism to enter and exit the cell, and that involves the use of vesicles. Endocytosis is the movement of large substances into the cells by the means of a vesicle, and exocytosis is the movement of material out of the cell by means of a vesicle. And the vesicles can be released to the outside of the cell. So here are these large particles over here, and actually we're going to start here. These large particles need to leave the cell. They're probably waste products, so we need to get them to leave the cell. So what happens is the cell is going to form a vesicle around them, and that vesicle is going to fuse with the cell membrane, and those substances will be released out of the cell. And here the opposite is happening. These large particles, maybe they're food, they need to move into the cell. So the cell will actually pinch and form this vesicle around some of these molecules, and then the vesicle will transport those materials into the cell. Here's another example summarizing the protein pumps, right? These require ATP, and then the formation of vesicles. Endocytosis, substances are going to get into the cell, and during think X as exit. During exocytosis, substances can move out of the cell. And again, these two processes, endocytosis and exocytosis, also need ATP. So I hope that was a helpful introduction to cellular transport. Thank you so much for listening.